give you a really quick recap. I had read some of the posts that you had started on the discussion board related to the content that we were covering in the last lecture. So just before we get going into what you need to know from today's lecture, one of the last topics that we stopped with in the last lecture was related to the lipid rafts. So the lipid rafts are these microdomains. They are little islands within the plasma membrane that have very unique properties. They have very specialized molecules. There are different types of lipid rafts. So there are flat ones, which we call planar lipid rafts. Then there are the ones that invaginate, which we call the calveoli. And we have different molecules there. One of the things that we know is at these lipid rafts are things like G protein coupled receptors. They're enriched at these lipid rafts. We have molecules like flotilin. We have calveolins. We have other things that make lipid rafts special in addition to the enrichment in cholesterol. But they represent these small little areas across the plasma membrane where we have unique properties and where we have unique molecules. Some of the other unique molecules that we talked about in the last lecture are things that are related to Alzheimer's disease. So one of the molecules that are in lipid rafts, not uniformly across all of the plasma membrane, seems to be things like amyloid precursor protein, which will be cut and then start to aggregate the A-beta peptide or the fragments that become toxic to neurons. And that's one of the reasons why we talk about these unique domains that are known as lipid rafts, and that's sort of what we ended off with. Today, we're going to be talking a little bit more about the plasma membrane, but in particular, we're looking at it generally as a plasma membrane, and embedded within the plasma membrane, we have these specialized proteins that are going to be really useful in our discussion. And this is sort of a different type of diagram that I'm going to show you later. It's the same idea that these plasma membrane proteins that work their way through the entire lipid bilayer are really the only way in which we can get the movement of charged particles or ions across this lipid bilayer. And we're going to look at some of the unique properties of these different plasma membrane proteins through today's lecture and carry this over into Monday's lecture. So as this diagram is showing you a little bit here, this plasma membrane is that fluid mosaic model that you learned about earlier. This is actually kind of interesting for you to think a little bit about. If molecules are moving, like you see in this diagram, and we have different types of channels and or receptors embedded within them, how come they don't move too far to the left or too far to the right, and how do we keep them in the areas that we have here? So again, just something for you to think a little bit about, because if you have been reading Nature or Science in the last 24 hours, there have been some really incredible papers around the GABA A receptor and new identification of different channels associated with GABA A, and these also move back and forth within the membrane. So just as an overview, this is that phospholipid bilayer that you've all learned about previously in many of your classes. And these channels, or these proteins in general, so channels are proteins, and in general, when we talk about how ions move across, they cannot go through the plasma membrane. They need help crossing it. And generally, we are talking about channels or receptors with channels embedded within them within this course. The main ones that we're interested in this course are things like sodium, potassium, calcium, and chloride. And I had mentioned to you that using only these four ions and using the movement of these four ions, we can actually encode for some very unique types of information. And it's pretty incredible when you think about it. Just opening and closing a channel to allow one of these four ions to move through it, allow you to differentiate between, by closing your eyes, touching a piece of wood versus touching metal, or being able to identify the letter A versus the letter C. All of that is because of these four different ions that are moving through different types of neurons and their associated channels that are embedded within them. So again, when we're talking about these ions, we know that this plasma membrane keeps these ions in separate compartments. We know that there is usually a gradient, which means that on one side of the membrane, there is a difference in concentration as opposed to the other. And as a result of this separation of charge, we have what is known as a resting membrane potential, which I'm sure you've learned about in previous courses. And the next series of lectures starting on Monday will go into a lot more detail about how we calculate it, how we can actually manipulate this resting membrane potential, and how this results. So again, by changing this resting membrane potential, by opening and closing ion channels, using one of those 4 different ions that I showed you earlier, we can actually generate changes or electrical signals that will result in some kind of signal being recognized by that neuron. And when we make the membrane potential less negative, so if it's sitting at minus 70 millivolts, for example, and we increase it or make it less negative and it gets to like minus 10 or 0 millivolts, then we say that we're depolarizing, or if we move it further away from resting membrane potentials, we say that we are hyperpolarizing. And again, it's just to orient all of you to make sure that we're all using the same vocabulary that we'll be talking about in more detail on Monday. So I don't also want you to get the impression that neurons and muscle cells or cardiac cells are the only places in which these ion channels are actually important. In fact, it turns out that if you were to take a look, any type of cell that you have within your body have pretty much the same systems that you have within the nervous system. So again, you don't need to worry about the details that are on here, but one of the things that I did sort of as I was leaving research to get into teaching was I was asked by the physiology department, I got a call, and they were trying to finish off a paper, and they wanted to know if, for example, pancreatic cells, do they secrete in the presence of a neurotransmitter like GABA? And could we actually measure GABAergic channels within pancreatic cells? And it turns out that most cells within your body actually have the same receptor systems that we're going to be looking at here in this course. So again, even in endocrine systems, outside of the classic neuron, muscular, cardiac systems that you have been exposed to, it turns out that most of what we'll talk about is actually applicable to every other system. As long as you have a membrane and you have proteins in that membrane, you're likely going to see the same mechanisms everywhere that you look. Okay, so if we're looking at these changes in the membrane potential, and this is how neurons signal, it's really important that we start off with a base, and these are really basic concepts, and I don't want you to feel like it's going to be a really easy course. It's going to start off very slowly to make sure that we're all on the same page, but then we're going to move into much more specific information that you need to be aware of, some of which will be coming up at the end of today's lecture. So I told you that the only way that ions can actually flow through from one side of the membrane to the other are through these embedded proteins. One of those different types of embedded proteins are channels. These are classic ways in which we can get ions to move across this membrane. And if you were to take a look, these ion channels that we see here always have sort of this typical cross-section. It doesn't matter what channel we're looking at. It's a very stereotypical ion channel that we have here. Remember, this is an aqueous environment, so this pore, this central area that you see here, and outlined for you right in the middle would be a representative ion. We have this aqueous-filled environment. Also associated with most channels, except for the ones that we'll talk about on Monday, those are not going to be exactly the same. Most of these channels have some kind of filter or some kind of gate that is associated with them. So here in this particular diagram, we have a part of the protein that juts out or sticks out that actually prevents ions from flowing through, and only flow through when there are specific conditions that are met. And that means that because of this selectivity filter or this gate, these different ion channels usually don't let every ion through. Only specific ions go through, and it will only open in response to specific signals. Again, there's no need to memorize numbers or anything else. You don't need to know the relative diameter or the length of any of these different channels. It's just for you to get a perspective on what is going on in this particular slide. So again, one of the key elements to everything that we're talking about is, one, starting on Monday, we're going to be talking about these concentration gradients that exist within neurons. And two, and very importantly, when we are talking about the movement of these ions across these different protein channels, it's always down their concentration gradient. So keep that in mind, that we need a concentration gradient. It has to exist. Normally in neurons, in cells, cardiac cells, etc., we have this concentration gradient. And so there's always a gradient in physiology that we can look at in normal circumstances. But keep in mind, it's always a passive movement. It's down their concentration gradient. And that means that your neurons, which are tremendous consumers of energy, so if your neurons had to use active transport to transport ions all the time, you actually would fall over within about one or 2 seconds. You wouldn't be able to actually have a functioning brain. The fact that it's using a passive mechanism actually is something that is very useful for the most energetic organ in your entire body. So again, the movement of this ion and the way that it flows into or out of a cell depends on the concentration gradient as well as the electrical potential across the membrane. So positive charges will be attracted to a negative environment, in other words, and negative charges will be attracted to a positive environment, just as you would expect. So do you have any questions on sort of the basics before we go on? No? Okay, so how do we open up this gate that is embedded within this channel to allow ions to flow through? So the first thing is we need to have a concentration gradient. So let's assume for a second we actually have a concentration gradient that will be at work. Then how do we actually open this gate to allow these ions to flow through? So similar to what you all experienced a few minutes ago, when you were trying to get into this auditorium, those gates would be represented by the doors at the back. And in fact, these are non-selective ion channels, if you think about it, because they're always open, and it doesn't matter who's going through. But we're talking about opening and closing these doors. How do we get this door to open, and then how do we allow either the movement of students in or the movement of students out? So the probability can be regulated by a number of different factors. It depends on the channel. So these are not going to work on every single channel, but it really depends on the channel that we're looking at. It could depend on the membrane potential. So we'll talk a little bit more about that on Monday. So some of these channels are voltage gated, so having a change in voltage might make the channel open. So it could depend on the membrane potential. It could also depend on whether or not a ligand is there. So it won't open in response to a change in membrane potential, but it might change in response to having a neurotransmitter present or a different drug present in order for it to be able to open. And it also depends on whether or not, even if all of those conditions are met. So we'll talk about this with the action potential, but many of you are already thinking, I know about voltage-gated inactivation. So even if there's a change in voltage, and even if the channel is open, there may be an inactivation gate, and so there might be a biophysical constraint where the channel is not capable of opening again within a certain period of time. And again, is there actually a gradient that's still left after all of this has occurred? So these are not necessarily all going to happen in the same channel, but these are different things that affect whether or not a channel can open or not. Do you have any questions about this before we go on? And we will be covering each of these in turn in the upcoming lectures. So I guess the big question, and one of the things that I'd like you to do as much as you can in this course, is to really question whether or not all of this is actually accurate. Because if you're thinking about what 10 nanometers represents, it's really hard to envision that is our model system, because I left you in the last lecture with this question, is our idea of the action potential actually correct? And there are some individuals that say, no, our idea of the action potential, the movement of ions, is actually not the whole story. It's actually the biophysical changes in the lipids in the membrane that actually convey information. That idea and challenging that idea is important. So we need to make sure that the theories that we're presenting to you are actually accurate. And this is one of those ways in which we can verify that when we talk about these membrane channels that span the membrane, Rod McKinnon who is shown here. And I kind of regret not going to his lab because I had a decision to make at the end of my PhD. Who do I go with? Do I go to New York and I go work with Rod McKinnon, who at the time was a really young up-and-comer. He was about to win the Nobel Prize. Or do I go somewhere warm? And unfortunately, because I was in Canada, I decided to do the wrong thing and I went to somewhere warm. I kind of regret not working with Rod because he's gone on to make some amazing discoveries. He did win the Nobel Prize for being able to elucidate the structure of these channels. And it turns out all those pictures that you see in your textbooks where we have the extracellular domain, the plasma membrane domain or the transmembrane domain that you see here, and the intracellular domain, when he does X-ray crystallography, it's exactly right. And in fact, all the theories that we've developed about embedded channels have been confirmed by not just Rod McKinnon, but many others to be exactly the way that we would propose them to be, and they've been validated through this X-ray crystallography. And again, that's just for your own information, and if you have the chance to work with Rod McKinnon, please take that opportunity. It's an amazing lab for you to be involved in. And sorry, I just gave him like a red nose. Okay, so going back to this idea of channel selectivity, there's a gate. There are some biophysical constraints. So that means that these different channels, we have a lot of different proteins that are embedded within a patch of membrane. And this is just a normal patch of plasma membrane. We're not even looking at lipid rafts. There might be a small area right beside this where a lipid raft microdomain might be existing. But just looking at a normal plasma membrane, we will have embedded within it many of these different types of proteins, and some of them will be voltage sensitive, some will be open channels, some of them might be responsive to different neurotransmitters. And when we look at them, we always have to look at whether or not they're selective or if they're not selective. For example, I told you the analogy earlier of the open door. So an open door, it doesn't matter. You don't have to be a certain size. You don't have to be a certain height. You can just walk through the open door as long as you fit through the door. And that would be a non-selective channel. That would be an example of something like a connexon. It allows anything of a certain molecular weight through. What we're going to be looking at through most of this course is that most of the channels indeed are selective, where we only, after opening, one ion will flow through, or at most two ions might be able to flow through at a time. And they can only flow through, again, if they are open and in a certain configuration. And again, going back, how do we know that everything that we tell you is actually accurate? A lot of these studies not just come from X-ray crystallography and the purification of membrane proteins, but using electron microscopy to be able to identify a patch of membrane. And here we're looking at it as an overview. We're looking down at the top of the channel. And you can actually see the acetylcholine receptors embedded within the membrane. So if we were looking down at a patch of membrane from above, we would actually be able to see the structure of these different acetylcholine receptors. So again, proving that our theories are actually well-founded. We can observe the same phenomenon that we are going to be talking about. So under normal circumstances, one of the things that we will see about these channels, so one, there has to be a concentration gradient for the ion to flow through. Two, they're usually gated, so it's not like they're open all the time. And it turns out because they are proteins, and because they're proteins in this fluid mosaic, so they move around, it turns out that under normal conditions, when they're not doing anything, they're just sitting there, they actually open and close spontaneously, and they close just as quickly. But there is sort of an opening and closing under normal conditions. But they're open and closing so in such a short time interval that it makes no biophysical nor physiological difference to the neuron. So one of the things that we're interested in is this value which is known as tau. And this idea of tau, this Greek letter tau here, and I've spelled it out for you. You might see it represented either way on your term test. We need to actually measure this mean open time. Like, how long is that channel open? So under normal circumstances, it opens and closes spontaneously, but it shuts just as quickly. We need to increase the mean open time, or the tau. How can we increase this value of tau? Do we need to add a drug? Do we need to change the membrane potential? And we need to actually see what sorts of conditions will increase this value of tau so that the probability of the channel being open increases, so that we know after a neurotransmitter has bound to the receptor, this value of tau will actually increase, because that will increase the probability that the channel is going to be open. Again, do you have any questions about this before we go on? No? Okay, so again, some of the other things that we can look at is when we open and close this channel, we talk about not just the length of time that this is open. So right now we could measure tau, it's open, and then it closes. So we would measure like how long is it going to stay in this state here. So we're now measuring tau. We're measuring the mean open time of this particular channel. And during the time that we are measuring this tau, we say that the channel is activated. We're measuring the open state of this particular channel. And we can also measure how long it is going to be staying closed. And this is the deactivation of this particular channel and the probability of the channel not being open. So again, pretty simple concepts. We're just using these as operational definitions. And you can see here, when we're taking a look, does this channel meet all of the criteria that I've mentioned to you before? So does it have specificity? Do you see more than one ion flowing through? So at least on this diagram, do you see more than one ion flowing through? It's honestly not a trick question. If there's only one ion, you should only be seeing sodium flowing through, and it is selective for sodium. So can it become activated? So here is where you need to be a little bit observant of what's sort of happening here. What do you think is the change that actually opens up this channel? So is there a ligand that binds to it? Is there something else that's happening that you can observe when it's open versus when it's closed? So there's a change in the charge across the membrane. So this is representing the membrane potential. And one of the things that happens is this is a voltage sensitive channel. So it's voltage dependent. And again, this is the change in voltage activates the channel, increasing the mean open time, or the probability that the channel will be open, and then it deactivates or it closes. So when we look at these different types of gating mechanisms, there's this voltage gated, which I went through very briefly on that previous slide. Some of these are mechanically gated, so these are stretch activated physically in your hand, for example. Some of the neurons in your hand have embedded within their plasma membrane channels which will only be open when you physically touch them, or if you physically stretch or compress them, versus others that are ligand gated, and these ligands can actually activate it either extracellularly, which is what you're traditionally used to seeing, or we can also see intracellular activation by other chemicals as well, and we'll talk very briefly about that. So again, when we're looking at these physical changes that activate this channel, the first one that's mentioned for you up at the top, under normal resting membrane conditions, and this is at rest, and we're going to go through this again in some more detail, and I know you've had it in your other physiology courses, either Bio 271 or PSL 300. Generally, the inside of the neuron is much more negative than the outside, which is how we represent it here. For a variety of reasons, or through a variety of different pathways, we can actually switch the membrane potential, have it more positive inside than it is outside. And this switch in the membrane potential, the difference across the membrane, will allow a physical change to occur within the channel. That gate opens, and this is now accessible to the ion that will flow down its concentration gradient. Okay, so this is an example of a physical change. It may not seem like a physical change, but it is a physical change that opens up this gate. And this one is a lot more intuitive in terms of physically opening a gate. This is where we are stretch activating. We typically see this on mechanical receptors or mechanoreceptors within your cutaneous or skin systems or sensory systems there. And this is where we physically will pull apart the membrane or pull on the membrane. And as a result, it will open up the receptor, allowing ions to be able to flow through. Any questions on this before we go on? I'm hoping it's pretty much a review, but again, just making sure we're all on the same page before we get into some more details on Monday. So the other classes are ligand binding. The ones that you're most familiar with are when we have a ligand, like a neurotransmitter, that will bind to the receptor and then allow it to open after the ligand has bound to it. So a classic neurotransmitter might be glutamate. So glutamate binds to the extracellular face of the channel protein, and as a result, binding will then physically change the conformation of the channel, and it will open, allowing for ions to flow through. We can also, for example, have intracellular activation. So some potassium channels, not shown on this particular diagram, some potassium channels, so not shown on this diagram would be the source of calcium, but you might, for example, have this potassium channel, which will be opened only in the presence of elevated intracellular calcium. So that calcium could come from a nearby calcium channel, which isn't shown on this diagram, or from an intracellular store of calcium, where the calcium builds up, will bind to the intracellular region of this receptor, and then allow the channel that's part of the receptor to be able to open up. So I just used two different terms there, a receptor and a channel. I kind of use those interchangeably. Typically, when we talk about a receptor, built into the receptor is this opening or this channel that is part of that receptor. So if it's confusing to you, please let me know. I'll be happy to try to explain that to you again in a different way. Any questions before we get going? No? Okay, so does a channel always become activated? And the next two slides beyond this one actually get into whether or not there's always activation. And it turns out that we don't always have to have ions flowing through when we have activation of that particular channel. So one of the things we've already talked about is inactivation. So if we have a continuous stream of action potentials, you'll already know that the voltage-gated channels will inactivate, so they can't open again. So even if we have the correct stimulus, they still remain inactivated or closed. So that's one example, the one that you're probably the most familiar with. The other case would be something like desensitization. If we have prolonged release of glutamate, or we have too much of a neurotransmitter around for too long a period of time, even though the stimulus is still there, that channel will not open as well as it did when the drug or the neurotransmitter was first applied. And then we have open channel blocks. And for those of you who have taken other neuroscience courses, you'll know, for example, that one glutamate receptor under resting conditions is normally blocked by magnesium. So this is the NMDA receptor. It's normally blocked by magnesium. And this occludes or blocks the pore. It's a lot like looking at a drain and having a stopper within the drain. You need to be able to remove the stopper in order to allow ions to flow through. And this open channel block, of which one case would be magnesium blocking an NMDA channel, and that's one example, which is really important when we study things like synaptic plasticity and LTP and LTD, where NMDA receptors play a big role. Understanding this block is important, but I'm going to show you two other examples of an open channel blocker beyond magnesium blocking an NMDA receptor channel. So here are three different examples of do ions always flow through when the channel is open? The answer is no, it does not always flow through. The channel could be inactivated, it could be desensitized, or it could be blocked even though the channel is still open. And the example I just showed you was magnesium. And now I'm going to show you two more examples of this third case, or the open channel block. Any questions before we go on? No? Many of you will have heard of TTX, and we're going to be using TTX an awful lot in this course, and you're going to hear an awful lot of TTX. And it is an example of a classic open channel blocker. So here, one of the reasons why TTX is really important, and I want you to hold on to this information. I'm sure you've heard it sometime in the past, but you'll need to know a lot of this in the upcoming weeks. This channel, tetrodotoxin or TTX, is really only an open channel blocker of voltage-gated sodium channels. And why is that important? Because starting on Monday, we're going to talk about sodium channels, but they're actually not blocked by TTX because TTX only works on voltage-gated sodium channels. And what we're going to be talking about are leak channels that are found within the plasma membrane. So leak sodium channels are not blocked by TTX, only these voltage-gated sodium channels. So this is an example of an open channel blocker. So you could have action potentials, and these action potentials work to open up voltage-gated sodium channels. They are physically opened by a change in voltage across the membrane. Sodium flows down its concentration gradient, allowing for a change in the membrane, allowing for some kind of signaling to exist. But if we were then to apply tetrodotoxin, it actually sits in the open channel. The channel is open, but tetrodotoxin is now sitting right in the middle of the pore, so no more sodium can actually flow through it. And this is an example of how we could block an action potential because TTX is blocking this pore from allowing sodium to go through. And again, the information on here is really not beyond what I've just told you. It's really for your information. You'll need to know about TTX as an open channel blocker or an example of an open channel blocker. If you're really interested, you can read up more about the bacteria that actually causes the toxin that resembles TTX that's normally found on pufferfish, but it's really just trivia for you in case you're interested. Or if you want to go out and have sushi, apparently there is one Japanese restaurant in Toronto that will serve this to you if you want to take a risk and risk having some tetrodotoxin with your sushi. I'm willing to go with you. If you want to take me up on that, we can have a little bit of a field trip. And again, this often causes paralysis, and it's this particular type of pufferfish or blowfish that typically has the bacteria that produces the toxin that will stop you from being able to breathe. That was the second example of an open channel block, and here is the third example of an open channel block. So this is also very, very specific to voltage-gated sodium channels. Okay, this does not work on leak channels. So when we start talking on Monday about leak sodium, leak potassium, neither tetrodotoxin nor lidocaine nor anything else will actually work in the same way as is shown on this particular diagram. And I'm going to take you through this diagram. I'm hoping that it's all up there for you. And some of this relates back to the very first picture on the first slide. And you don't need to know all of the details, but I'm going to take you through a couple of things that are on this diagram. Whenever you are talking about a local anesthetic, so whenever you go to the dentist and you don't want to feel pain, you want to block those action potentials from traveling into your brain, one of the things that they apply is a local anesthetic. So they will target the nerve that is supplying your tooth, and they will inject this local anesthetic. And then you can't feel anything, and that nerve is being blocked by this open channel blocker. So nothing else is going on except the voltage-gated channels, when they open, they can't actually transmit anything because no sodium will flow through them. And I'm hoping this diagram will explain that to you a little bit. So any type of local anesthetic that we use that's an injectable local anesthetic always ends with a cane. So lidocaine, for example, procaine. Cocaine is actually an anesthetic, not something that you should use, but it is something that is clinically used by individuals that need to locally anesthetize a particular area of the body. And all of these local anesthetics that are always something cane, work in exactly this manner. They are all open channel blockers, all voltage-gated sodium channel open blockers. But we really don't understand the mechanism. Surprisingly, we have a number of drugs and derivatives of these drugs that you'll learn about in maybe some of your pharmacology, toxicology courses, but the mechanism isn't really clear on how they all work. So the one thing we do know is that they have to sit and block a voltage-gated sodium channel. So cocaine, for example, the stereotypical type of local anesthetic, will enter into a voltage-gated sodium channel, which is outlined for you here, and it will sit within the pore, and it will actually physically prevent sodium from flowing down its concentration gradient. But we don't know if cocaine actually does it as a charged particle directly, or because if you look at the structure of any of the local anesthetics, they are all lipophilic. That means that they can cross the plasma membrane. So when we inject in a local anesthetic outside of the nerve, it will then be able to cross into, pass through the plasma membrane directly, and then it might go intracellularly into the intracellular space because it is normally lipophilic. And then inside the intracellular domain, it could become charged as a result, and it may go in and block this channel from the inside region. And that's one theory. And another theory is that it might actually somehow, by a mechanism we still don't understand, actually become charged as it enters into the plasma membrane. In either case, whatever theory is correct, and beyond that you don't need to know any of those details, these local anesthetics block these voltage-gated sodium channels, which means that ultimately no action potentials can be generated. So this is the third example of an open channel blocker. And here you can understand why it's considered an open channel blocker. This voltage-gated sodium channel is still open. It's in its open conformation. It is still technically activated, but you can't actually get sodium to flow down its concentration gradient. So this is, again, a third example, all of them working in the same way, of an open channel blocker. The channel itself is technically still open, but we don't see the result of any ions flowing through. Do you have any questions on any of these different open channel blockers or anything that we've covered before we go on? No? Okay, so I know this is really terrible because this is like week one, and I'm trying to get you to think about the very end of January. But I want you to start thinking about how you can get, yes, you have a question.
Yes, on the previous slide, it showed that cocaine went from the intracellular compartment to block the sodium channel. Can it also go from the extracellular compartment to block the sodium channel?
So again, there's a lot of controversy about this. So if you're to look at the literature, these are the two main theories. In fact, there are a number of other theories that have been proposed. All local anesthetics, though, begin as non-charged particles. We can't actually inject things that will block, that are positively charged or negatively charged. So these are neutral lipophilic molecules. In theory, some people have suggested that there are other enzymes that exist in the extracellular space that could theoretically block from the outside, but it's not a major theory. Okay, so again, when we're looking at, this is still the same principle. Nothing charged can actually cross the membrane directly. This is a lipophilic molecule. If it were a charged particle, it actually would only be able to go through one of these channels. But so far, there's been little evidence to suggest that this mechanism of having a charged molecule directly from the outside blocking, there isn't as much evidence as compared to these two mechanisms here. Okay? And a great place to go and learn more about this is to finish your undergrad, get into pharmacy. You'll learn a lot more about this in your second year pharmacy classes. So something to maybe look forward for some of you. Any other questions? Yes, at the back. Are all the caines lipophilic? The vast majority of them are indeed lipophilic. Not all of them, but the vast majority. It's a good question. So things like xylocaine, if you're in research, you'll often inject xylocaine, which is a local anesthetic. That is a non-charged lipophilic molecule. Procaine, lidocaine, many of them are. Okay, so again, just a little bit of an overview of what you need to start preparing for. I think some of you have already begun the process of forming study groups. I'm willing to join any study groups that get set up on Thursday or Friday. Usually they're about 1 hour long-ish, and I want you just to be able to ask questions, and I'll be able to answer any of those questions directly. So if you do happen to have set up a study group, wherever you're meeting, just let me know. I'll be happy to show up, ask you questions, answer any questions that you might have. And again, just to make sure that we're all on the same page, this is actually from Monday's lecture, but you should be able to answer